Without genetic diversity, evolution halts and species eventually go extinct.
It's among the lowest diversities that we have observed in any butterfly so far.
Human activities often destroy habitats, making the populations more and more isolated from one another. This is what Niclas and I are focused on. What happens with the genetics in small populations?
What's the importance of genetics in local adaptation?
Genetic diversity is the basis for the ability to adapt to changing circumstances and changing environments. So without genetic diversity, evolution halts and species eventually go extinct. If they can't evolve and track changes, then life stops basically. So that's why genetic diversity is really, it's key to life as such, basically.
In a changing environment, yes.
Yes.
Yeah. Mm.
Mm. And we know that all environments do change. I mean, there are very few environments that stay exactly the same.
So genetic diversity is important, but what is it? Well, within a species, individuals can have genes with different variations. These variations are called alleles. If there's a lot of these variations within a species, there's a high genetic diversity, and therefore a higher chance of having suitable genes if the environment changes. Another way to phrase genetic diversity is the lack of genetic problems like inbreeding and genetic drift, but more on that later. So why does a species lose genetic diversity?
The general explanation for why species become rare and why they go extinct locally is habitat fragmentation. So human activities often destroy habitats, making the populations more and more isolated from one another and broken up into isolated subpopulations. So that's what we call fragmented populations, basically. And this is what Niclas and I are focused on, the genetics of what happens with the genetics in small populations.
So has that happened with the Apollo butterfly? Loss of genetic variation?
I mean, the results we got from the initial study shows that it has very low genetic diversity compared to other butterflies. So it's among the lowest diversities that we have observed in any butterfly so far. So that seems to be a consequence of the population decline. This has also led to kind of genetic differences between the populations. So they have, since they are so small, the alleles have drifted in the different populations, so they have accumulated genetic differences over time. So that means that they are not identical anymore.
No, since they are not exchanging individuals, it's too far away between these subpopulations. They start to evolve independently of one another and not exchanging any individuals. And that has consequences.
Small populations tend to lose genetic variation due to a process known as genetic drift. On top of that, individuals in small populations are more likely to mate with close relatives, simply because there are fewer options. This leads to inbreeding, which can be a problem especially when harmful mutations are involved. These are called recessive deleterious mutations.
The mutations that are bad for you, but they are masked by other mutations because they don't come to expression since they are recessive.
As inbreeding increases, so does homozygosity, meaning individuals are more likely to inherit the same allele from both parents. And that increases the chance that these harmful recessive mutations will be expressed.
That's a process called inbreeding depression. That the inbred individuals have lower fitness than an outbred individual. And these processes are more prevalent in small populations than in large populations.
I think we're also both quite interested in the evolutionary aspect of this, when these things happen to a species. How does that affect genetic diversity, accumulation of bad mutations, and so on, when you have this fragmentation and limited gene flow between populations.
And Sweden is also a very interesting part of the world to study these aspects, because 20,000 years ago there was almost no life here. It was just ice. So Sweden was recolonized since about 10,000 years ago. So all species have an invasion history, and that invasion history has affected many of the parameters that are affected by humans now. So we have to try to distinguish between historical processes that affect genetic diversity and contemporary processes that affect genetic diversity.
We didn't know that from start, but it turned out that from our first study there that the origin of these populations in the northern part of Sweden and southern and central part of Sweden, they actually came to Sweden from two different routes. Medelpad, they are more related to Finnish and Eastern European populations, while the southern Swedish populations are more related to Central European populations. So it seems like they colonized Sweden from two different routes.
So I think this is one of the rarer exceptions where we have a genomic genetic study to back up and advise on the conservation plans.
So I think our aim was to use kind of a genomics approach to make informed decisions for the stakeholders that they can use and in direct conservation practices.
It's a tool that is gaining more and more application, and I think that we can, as geneticists and conservation geneticists, we can provide a lot of information that is useful for the conservation projects.
So we can take the cloud Apollo as an example of that, how the results can be used in direct actions for conservation. So now we know, for example, that we have population structure in Sweden, and we also see some evidence maybe of local adaptation. So now we're actually directly testing if it's a good thing or a bad thing to mix populations. So we can breed them in captivity, and then we can measure fitness of the offspring when we cross individuals from the different populations, and then we can see, do we see evidence of this outbreeding depression, which is a consequence then of you have different populations that are locally adapted, and then if you mix them, the hybrids might become less adapted to the environment than the parental populations. So that's a risk. We think that's a low risk in this case, because we think it's more important to restore the level of genetic diversity and the adaptive potential of populations by mixing individuals from different populations rather than maintaining them as separate units, let's say. So that's a couple of direct insights that you get from genomic data that you would never have otherwise. I think another thing that you get from genomic data is you can estimate the effects of inbreeding. So
that's very important.
Yeah.
So now different analytical tools how to estimate the level of inbreeding in populations, and even at the individual level, so we can say that this individual is inbred, this one is not.
So we've done this in a stepwise manner because we didn't have access to a reference genome for the clouded Apollo when we started. So we only used Illumina short-read sequencing for these populations, and then we mapped it to the Apollo butterflies, which is a relative, but it's kind of a distant relative. It's within the same genus, but it's still quite far away, evolutionarily speaking. So that means we could estimate genetic diversity. We could look at population structure within Sweden and see that they are differentiated and so on. So from that we learned then that we have these different colonization routes to Sweden, we learned that they have very low genetic diversity compared to other butterflies, that we have some structure, that populations within areas are not identical, so to say. They accumulate differences due to genetic drift. But then we took this next step, so we decided to make a reference genome for the clouded Apollo to have as a tool for forthcoming analysis, which can be more detailed. And there we got that into the ERGA pilot, the European Reference Genome Atlas pilot set of species. So there we actually combined long-read sequencing with PacBio. We used high-C contact maps and RNA-seq, so we could get a very, very good quality genome assembly in the end from that. So from that we learned that they have huge genomes compared to other butterflies. So in general, butterflies have a genome that's around a few hundred megabases long, let's say. But the clouded Apollo and the Apollo, they have around 1.5 gigabase long genomes. And this has been a consequence then of that they have accumulated a lot of these so-called transposable elements, which is a type of repeat that spreads by itself in the genome. So in the clouded Apollo, it's more than 70% of these selfish genetic elements called transposable elements that constitute the genome. We also learned that the chromosome structure is pretty conserved in the clouded Apollo compared to the ancestral Lepidopteran karyotype, so that means that they more or less have the same set of chromosomes as most butterflies have. But they also have a fusion between an autosome and one of the sex chromosomes. So they have one chromosome fewer than the Apollo, for example. So that's also something we learned from that. Only the genome assembly, so to say. But I think the most important thing is that we can actually now do follow-up analysis. So we can, for example, pinpoint locations in the genome where populations differ, for example. And then we can try to assess if this is a consequence of local adaptation or random effects.
And we can actually get to the genes that where these populations segregate. So we can say that it's in this gene or this genomic region or this chromosome and so on.
And we can also look at all the individuals and see where are they homozygous because of this inbreeding effect. So we can actually make a detailed map of the entire genome for each individual and say, now look, these are very inbred because they are homozygous over large stretches of the chromosomes. That's something we couldn't do with a reference genome from a relative.
Remember Martin Elmestål, who was out counting butterflies earlier in this series? He says that flying butterflies have been seen at 2 locations where they were released. That's the first generation to be created entirely in the wild from butterflies or caterpillars released in previous years. Will it work? We won't know for a few years as the populations are still small and sensitive, but there are hope for the clouded Apollo butterfly.